All right, well, next up we have me. So hi, everybody. I'm Will Samuel. I'm a master's student at the University of Alaska Fairbanks, and today I'll be discussing my master's thesis entitled When Beavers Get Burned, Do Fish Get Fried? Assessing beaver effects on fish in a fire-dominated ecosystem using eDNA. So wildfires are increasing in size, frequency, and severity in boreal North America. Over here on the right, you can see the distribution of the boreal biome in green, and the distribution of wildfires in Alaska since 1984. Those are in orange. And note that those two distributions line up really well. And that's because wildfires are the dominant disturbance process in a boreal biome. Like I said, wildfires are increasing in size, frequency, and severity. And you can see that not only have wildfires increased in frequency, but also the cumulative total acres burned in the past couple of decades. It's important to consider that beavers and wildfires have the potential to interact with each other. So a dissertation published from Emily Fairfax suggests that beavers actually have the ability to buffer wildfire effects on streams. And they basically do this by damming the streams, flooding the riparian vegetation around that, and increasing the distance between the stream channel and the wildfire. Kind of looking at that from the other direction, wildfires have been shown to control beaver populations. So this study in a national park showed that as the cumulative area burned increased, the number of active beaver lodges, or a proxy for the beaver population, decreased. And a follow-up study 10 years after that showed that that population did not recover, and they mostly attributed that to the high severity of the wildfires, the fires that burned really hot. And it's important to consider that both wildfires and beavers can affect fish in numerous and complex ways, depending on the local environment. And these relationships can be further confounded when beavers and wildfires are potentially interacting with each other. So my thesis kind of comes at the center of all of these topics, and it kind of comes to one question, which is how do beavers mediate the effects of wildfire on fish? So the first part of my thesis is looking at how do fires affect beavers. That was kind of a GIS study, and I talked about that last year in case any of you caught it. The second part of my thesis is looking at how do beavers affect fish, and that's what I'll be focusing on today. So I'm conducting this research in interior Alaska. This gold star right in the center of the map is Fairbanks, where we are today. And I'm doing this research in 5 large river basins you can see on the map. This is a really large study area, 21,000 square kilometers. So this is about the size of Hawaii, Massachusetts, New Jersey, or Connecticut. And about 25% of this has been burned since 1984. Again, stressing that it's a fire-dominated ecosystem. You can see all the colored polygons here represent wildfires that happened at different times in history. And I also want to bring your attention to Caribou Poker Creek, which is a long-term ecological research site that the University of Alaska owns. So my lab has been conducting fish surveys here for the past couple of years, and I'm going to be coming back to this later in the presentation. So I just kind of wanted to orient you guys to this and kind of let you know that this is a good representative tributary to conduct research in, because it's similar to a lot of other tributaries in the study area. Briefly touching on my study species, Arctic Grayling. This is a widespread fish throughout North America. You can see its distribution in purple there. And it's mostly an important sport fish, but there is some subsistence use. And they require accessibility to headwater and tributary streams for feeding, rearing, and spawning. And briefly touching on one of the tools I'm going to be using to ask these questions, which is environmental DNA, or eDNA. If you're not familiar with this, this is basically particles of DNA that shed from an organism into its environment. So in this case, it's pieces of DNA coming off of the fish's skin, slime, or excreted as fecal matter, and they get deposited and stay suspended in the stream. It can persist from anywhere from 24 hours up to some unknown amount of time, depending on the environmental conditions. It's been shown to be transported by flow about 1 kilometer. And degradation is the main limiting factor for transportation in streams. And that can be affected by a variety of environmental conditions, things like water temperature, UV radiation, microbial activity, pH, and a variety of unknown factors as well. And importantly for my study, eDNA has been used to estimate fish abundance, but it's still a rapidly emerging field, and there's a lot of questions and assumptions that haven't been figured out yet. So this part of my thesis kind of breaks out into 2 main objectives. First, I hope to estimate the Arctic grayling distribution and abundance across tributaries using eDNA. And once I have those estimates, I'll compare those results to beaver distributions and wildfire history that I summarized in my first chapter. So for that, I'm going to start by trying to develop an eDNA catch per unit effort model. Here we're hoping to establish some sort of relationship between the number of fish in the stream and the concentration of eDNA. So for this, I sampled 10 sites, which I'm calling intensive sites. This just means that I sampled them multiple times throughout the summer, and I sampled fish and eDNA there, and that's what I'm using to build my model. And then if that model turns out well, if there's a good relationship, I'll use it to estimate fish abundance across these other sites that I sampled. So I'm calling those broad sites, but there's these 56 other sites that I sampled once each, and I only sampled eDNA at those sites. As far as the methods for this, I used a combination of angling and electrofishing, and then I calculated a catch per unit effort, which was also standardized by stream area. And you can see it took some creativity to get all of our sampling gear hiked out to our sampling sites this summer. The eDNA essentially included filtering water. So we collected 3 replicates per site. You can see the filters down there in the bottom of the screen. And we also collected environmental parameters, things like stream velocity, temperature, pH, turbidity, specific conductance, and dissolved oxygen as covariates in the model. We folded the filters and stored them in desiccant. And then once we got back to the lab that day, we stored them in a -80°C freezer. I sent these out to a company called Jonah Ventures to process them. So they essentially extracted the DNA from the filter, they incubate or amplify it, and by doing that, they can quantify the amount of DNA. So the number of copies of DNA per filter. And then when I got that data back, I standardized it by a concentration, so number of copies per liter, a concentration in the stream, and then I averaged those replicate values for each site and sampling event. And that's what I've been using to build my models. So here's the raw data. It's pretty ugly. Once you log correct both the predictor and response, it looks a little bit better, but overall we ended up with an adjusted R-squared of 0.33. And those habitat covariates that I mentioned, things like velocity, stream temperature, pH, those did help explain some extra variation. Overall, I'm not totally surprised by these results. There was a paper published by Adam Sepulveda in 2020 attempting to do something very similar, and our results are almost identical. So my whole thesis wasn't hinging on this. There is this other body of research that shows really good relationships between eDNA. So I'm not totally done investigating this, but now I'd like to talk about why I think this relationship isn't better. And this is kind of my failure in fisheries, and not only for my project, but it's a broader challenge that fisheries and people using eDNA have been working on for quite a while to try to figure out if we can use eDNA to estimate fish abundance. So to think about why our model turned out how it did, I'd like to orient you back to Caribou Poker Creek. And again, we've been conducting fish surveys. So in 2022, this is what the fish distribution looked like. These little red marks are the very upper extent in Caribou Poker Creek where we observed fish. And then the blue is everywhere where we caught fish in between. And I'd like to focus in on this area where we also sampled eDNA. So this is what the eDNA values look like at our 4 eDNA sites in Caribou Poker Creek. Their size is representative of the concentration. So what you see is as you start at the top of the tributary, the eDNA concentration is relatively low. And as you move downstream, it appears to accumulate or magnify. And this would kind of explain if eDNA really is accumulating as it moves downstream, this would explain why my model relationship isn't good. And the past research using similar methods wasn't good because our fish surveys were on a much smaller scale. Our fish sampling reaches were only 250 to 500 meters. And if the eDNA is representing a much greater portion of the tributary, that might explain why the relationship between these aren't good. And you could say that this eDNA concentration is just a function of the fish distribution in Caribou Poker Creek, but we sampled fish there, so we can also plot those values. Again, here the size is representing the catch per unit effort, and this is standardized by stream area, so it should account for that. And what you can see is that catch per unit effort is pretty uniform throughout Caribou Poker Creek, and it doesn't really follow this trend of eDNA, which seems to accumulate as you move downstream. There's one exception, which is this site over in Caribou Creek, which is kind of an interesting spot where the fish get concentrated beneath a beaver pond, so they're really easy to catch. So I think that's partially artificially inflated catch per unit effort, but I also think that high fish concentration is represented over here in Caribou Creek at this eDNA sampling site. It's a little hard to see, but that eDNA concentration is almost twice that of Poker Creek. So I'd kind of like to revisit the assumptions that I introduced this presentation with, thinking about eDNA persisting in an environment for 24 hours or somewhere around there, and the idea that eDNA can travel 1 kilometer in streams or less. At least that's what the scientific literature has shown. So in Caribou Poker Creek, we know the most upstream point where we've caught a fish, which is that gold star up there. It's about 8,000 meters, 8 kilometers along the stream channel to the bottom eDNA site. And we measured velocity while we were sampling eDNA. So if you average all the sites, all the sampling events, we came up with an average flow rate of 0.34 meters per second. So we have a distance and we have a rate of travel. We should be able to calculate how long that eDNA should take to travel from the very top of the tributary to the very bottom, or in other words, a residence time of eDNA in the stream. And when you do that math, it comes out to 392 minutes or 6.5 hours. That's really quick, surprisingly quick. So this kind of agrees with our first assumption that that eDNA persisting for about 24 hours, if that is the case, then we can probably detect it at the bottom of this tributary. And but this other assumption that eDNA only travels 1 kilometer in streams, it seems like we're kind of violating that. So I'd kind of like to go back and update that assumption and say that as long as the eDNA doesn't settle and degrade in 6.5 hours, or maybe 8 or 10 hours in a longer tributary, that it can travel much further than this previous literature suggests, maybe 5, 10 kilometers, maybe even further than that. And that I think that the reason for this is because of our environment, that we're at these high latitudes where we have cold stream temperatures, the angle of the sun is really low, so solar radiation is lower here. It has to travel through more atmosphere before reaching the stream, and our streams are just generally less productive, so there's less microbial activity that could degrade that DNA. As far as next steps for this project, I still need to predict out across those other 56 tributaries. So I'm comparing different model structures for that. I kind of already talked about the linear model or GLM, and how that may or may not be useful. I'm not totally done playing with that, but an R-squared of 0.34 is only so useful. So I'm kind of looking at alternatives like an N mixture model or a multi-state occupancy model, which fits the eDNA data set really well and kind of gives you categorical abundances, high, low, medium abundance across this whole study area. And then I've also considered other models like a Bayesian model with allometric scaling. I'm not sure what I'm going to do with that. I think in an ideal world, I would design a couple different models and do what's called ensembling them or model averaging and see if they all agree. If they all have the same general trend lines in predicting fish abundance across this study area, I think we can be somewhat confident in our results. And then the final step to connect that back to my thesis and questions is to summarize the habitat factors that affect fish abundance. So mostly beaver distribution and wildfire history. So in my first chapter, I summarized I surveyed beaver ponds throughout my entire study area. So you can see those black points here. And then the wildfires are represented by red. So we can look at sites with recent wildfires and high and low beaver density and see whether those have any correlation with the fish abundance derived from eDNA. With that, I'd like to thank everybody who contributed to this project, my committee and co-authors, especially my advisor, Jeff Falke, my technicians who helped last spring and last summer, all the volunteers who I could not have completed this work without, especially the hard summer field work last year. And I'd like to thank funding from the Strategic Environmental Research and Development Program and the Alaska EPSCoR program. And with that, I'd be happy to take any questions. I had looked at that. The reason, and I didn't do it, it's pretty expensive to run the samples, especially since I sent them out to a company. So I had to, I was paying for somebody else to do it. And the reason I didn't include that is because I thought that there would be beavers in pretty much every stream. And really what I'm looking at is beaver dams and ponds, which are the barriers to fish or potential habitat to fish. And so I'm not so interested in the actual number, the beaver population, but instead the proxy of that, which is the number of beaver ponds. Yep. So if you see a situation like your lowest. Yeah, so that's the goal. So with those 56 broad sites, I sampled at the bottom of the tributary, and that's the goal. Ideally, it's representative of maybe not the entire tributary, but a good portion of it and a good portion of the available fish habitat. So it's an assumption I'm going to have to make, and it's just because the eDNA world doesn't have answers for that yet. But yeah, that's the idea. What pore size are you using on your filter? 1.5 micron. And I have a 45 millimeter filter, and I was going for 5 liters per filter, if I could get that with the sediment. Yep. Yeah, so a lot filtering a lot of water. Cool. All right. Well, thank you, everybody.